Great. Well, thank you everybody again for joining us. It's been a very exciting day full of awesome science. And again, I want to take this opportunity to talk about some of our current work. And I think by now it's a very clearly recurrent theme that many of us in the room and in the field are interested in understanding why cells that are exposed to the same perturbation, in our case viral infection, actually have very different responses. We think that by starting to understand and explore how cells process information during infection, we can decode cell states and cell state transitions that are going to reveal molecular mechanisms behind pathogenesis, and also are going to help us unlock targets that will lead to the modulation of infection outcomes. We are parsing this big question into three more, not less complicated, but three smaller questions. We're trying to understand first what initiates a response. Basically, what are the sensors that are going to lead a cell to launch a response to infection? Once this cell is kind of going down the hill of these cellular states, how are those cell states maintained? Basically, we want to define the pathways and the checkpoints that are going to push cells into different functional states. And ultimately, we want to know what are the modulators that are going to help determine the fate. What is going to, once the cell goes into this cell state, what determines that one cell becomes resistant or clears infection or becomes abortive, as we have seen several times today, or ultimately succumbs to infection? So to do this, what we're doing is addressing this question from a multimodal way. So what we are looking at is the remodeling of cellular responses in a high-temporal resolution. In our case, the model is going to be A549s that are infected either with Zika or dengue. And according to the poll, I don't even have to say why dengue is important, since it seems to be one of the most favorite viruses in this audience. So, but again, these are highly medically relevant viruses that cause a very high burden of disease across the globe. So some of the metrics that we are using in this model of infection, again, A549s infected with Zika or dengue, are transcriptomics, both at bulk at single cell resolution, as well as proteomics, bulk, and organolepid, that I'm going to tell you a little bit about in a couple of slides. Both of these we're trying to collect as high temporal resolution as we can. So usually we do about 24 to 36 hour time courses, collecting time points every 3 hours. So this is the closest and the highest resolution that we can get at least in our team by now. We're also integrating phenomics, which is basically going to be live imaging using label-free and endogenous reporters of infection, and also cell states that I'm going to tell you more in a moment. I am not going to spend much time talking about, let's see, which is this, no, backwards, the other one. I'm not going to spend much time talking about where is the, okay, let's do it here. The transcriptomics and the proteomics. We have today one of the posters by the team from Karen Cheng, who's going to tell you more about this infectomics project. But just to give you a little bit of a teaser of the kind of information that we're getting, these are UMAPs of single-cell transcriptomics, where we have aggregated cells or infected cells by Zika and dengue, and we can identify three populations of interest. First, I'm showing you here the ones that are infected, where we can detect viral transcripts, and we see very low innate immune responses. We also have bystander cells that we have heard from other speakers today, where we cannot detect viral transcripts, but we do see a response to infection, in this case, innate immune responses. And one of the most interesting populations that we have captured and that we are able to now start investigating a little bit in more detail is this tiny population right here, where we can detect viral transcripts. They have a very high innate immune response, and they have very high signatures, transcriptional signatures of stress responses, and we call these ones hyper-responders. And even though they are a small group, we think that they do have a very important role in the population regulation to infection, to responses of infection. So right now what we're doing is trying to isolate these three populations here to characterize them better. But again, visit Karen's poster later today to learn more about this infectomics project. But what I want to focus for the rest of my time here is this phenomics project, where we are asking when and how do organelles change during infection. And we also want to see if we can capture enough information by microscopy that is going to allow us to identify infected cells based solely on their morphology. And this is a close collaboration with Shalin Mehta and the computational microscopy team at the Biohub. And one thing that I want to take a pause and highlight is that neither one of us, neither Shalin or I, could have done this individually. And really, by joining our expertise and joining our teams, we have been able to make this project work really fast and move very efficiently. I think that it's very important to highlight that communication, open communication, and constant communication is what is helping our rapid iteration. One, we define the biological questions. We talk to Shalin to see which are the best tools to answer those questions. We design the experiments together. We analyze the data together. And then we iterate very quickly to generate the best data that is going to serve our models and give them true predictive power. So I just want to highlight that this is, again, I hope the future of many other collaborations where we have these very dynamic interactions between multidisciplinary teams. But going back to the biological question, so how do we capture organelles and their dynamics? So here we're taking a page from Manu Leonetti's playbook. So this is a method that they developed a couple of years ago in which we can introduce endogenous tags into proteins of interest. In our case, we're introducing a GFP and HA tag into proteins that are prototypical markers of different subcellular compartments. The introduction of these endogenous tags allow us to track the behaviors of some of these proteins and some of these organelles and subcellular compartments without the need for overexpression or stains that can be short-lived. But even more, going beyond the imaging, we can also use this HA tag to capture these proteins, and using mass spec, we can look at the complex that they are in and how they are remodeled during different cell states. So using this method, we generated what we call the organelle box. And this is a panel of cell lines in two different backgrounds, A549s and 293s. Our group developed the A549s, Manu's team developed the 293s. For the A549s, we have 23 individually tagged cell lines that represent 17 different compartments. And we are sharing now this with the community as an open source, and what you will get is a little box like this with a little cute sticker. So if you're interested, you can scan the QR code and we can learn more. You can learn more about that, or just come and contact me after the talk. Besides the panel of cells that will come packaged like this, we also developed a portal in collaboration with CCI and many others here at Biohub, where we report this resource. We'll tell you more about the cell lines that we're generating right now. We give you all the QC images. We are also giving you some protocols of interest, how to label these cells, how to do organelle IP. And in the future, we're going to also include here data and some of the models that Shalin has developed so that you can analyze the data as well. So again, don't worry if you cannot scan the QR code right now. I'll have it at the end. So how are we using these cells to track organelle remodel during infection? So we took a subset of these cells that I just showed you, and we introduced a sensor for viral infection. So this is a translocation reporter. This is just an mCherry protein that goes from the ER to the nucleus after being processed by the viral protease. This is a design from Felix Baumeier and Mika Cortisi. Again, specific for Zika or dengue. And we're imaging these cells in a time course of about 20 to 36 hours, taking 30-minute snapshots. And we're doing fluorescence and label-free imaging. And these are some examples of the movies that we're generating where you see the infection sensor in magenta. Again, you're going to see that it moves to the nucleus as infection progresses. And we have here stress granules, mitochondria, and endoplasmic reticulum. So you can see clearly the remodeling of these organelles as a consequence of infection. So if you're like me, you're probably going to be staring at these images for a long time, trying to come up with some patterns or find something that is happening. But this is first bias, second, my eyes are not that good anymore, and third, is really not efficient. So Shalin's group developed DynaClear, which is a self-supervised method that maps 3D multi-channel time-resolved images into a lower dimensional embedding space where each cell is a dot. You can think about this embedding space as a blank canvas. And what we're doing is just posting pictures of cells individually that look similar. So for example, cells with fragmented mitochondria will go here. Cells with intact mitochondria will go here. And the distance between the dots, between the cells, either in this group or this group, reflect biological principles and biological relationships between these different dots. Simplifying these images and having them now in this lower dimensional embedding space allows us to perform some downstream tasks, including the robust classification of infection state based on our sensor with minimal human input. We can also then transfer that information into our label-free images to be enabled to predict infection states just by label-free without the need of any fluorescent marker, and that's something that we're working on right now with some level of success. And also we can align these cells and these images in time, and I'm going to show you an example of that in a moment. This is just an example of the embedding space that I was just talking about. So these are fate maps where you can see cells here in a non-infected state in blue, and as the infection progresses, you can see them move to this area right here that is appearing right now in orange. These are annotated based on the embedding space, not on the images themselves, again, showing that this approach is working. Similarly, we can do the same for organelle remodeling. So here, the uninfected cells are going to go from, this is ER, by the way, so these are going to go from this space to an area right here, again, reflecting the change in the ER that we know happens during flavivirus infection. Using this model, we can also, again, align the cells according to different conditions. Here I'm showing you two of those, one in silico alignment according to cell cycle, and the other one according to the infection cycle. So it's important to remember that these infections are asynchronous, as we were just seeing by Sane, that things happen at different times. But for us, that doesn't matter. If we can align them all in time based on the translocation reporter, then we can start comparing how different organelles are changing related to each other. And this is using dynamic time warping, inspired by the work of Alan Lowe, which is one of our colleagues at CCI. So clearly from what I have shown you, the models are learning something that is different about these cell states, about infection, how cells are moving and changing around, but it's something that we cannot interpret. So right now what we're doing and what Shannon's group is doing is helping us interpret what are the changes and how are these cells being separated into these different groups. So here I'm showing you the embedding space, and we're just showing you here basically the extreme of the axis, where you can see that these cells are definitely different in their morphology, and that's how they are being separated in the embedding space. Shannon's team is now engineering some features that allow us to really say, okay, what does this mean? What is the phenotype that we're looking at here? So for example, in this principal component analysis, we can see that for this group, the fluorescence intensity, the radial intensity is what is defining the features here. So this is something that is ongoing work right now, but the idea is that we're going to be able to then put a name to these changes that we're seeing in the different cells. And putting all of these together, here I'm showing you the dynamic remodeling of stress granules in uninfected cells, cells infected with Zika virus, or cells infected with dengue. And we have the embedded spaces here for the stress granules formation and here for the infection. And this is analysis that is ongoing right now, but what I can tell you is using these images, we're already starting to see a difference in the kinetics of formation of stress granules between Zika and dengue. And the idea is that we're going to continue doing this with all the cell lines that I just showed you in the A549 background. So where are we going to go from here? Well, we want to finish imaging all these cell lines. It's a huge effort, but we already have 3 different organelles that we are modeling right now, and the idea is that we continue this process with the 23 cell lines that I mentioned. Of course, we want to integrate this imaging data with other modalities. I told you that we are generating this high-temporal resolution transcriptome and proteome, and we would be happy to talk to some of you that are also interested in addressing this integration issue. Manu's team is starting to integrate these reporters in optical pool screens, and one of the things that we're really interested right now is we have this transcriptome and proteome and imaging data, but they're all from separate cells. They're not paired. So we want to try to get some paired measures that are really going to allow us to link a transcriptome, for example, with an image from exactly the same cell. And seeing that we are at the Biohub and zebrafish are one of our favorite models, of course, I mean, we have some examples here in the audience, we would love to develop some functional reporters and some of these organelle reporters in the fish that would allow us to track these different changes and this remodeling at an organismal level. With that, I want to finish and thank everybody. I invite you to visit all of our posters in many different things, the infectomics project that I talked about, but many other things that are going on in our group. Thanks to Manu's group that has helped us with the cell engineering. And here is again, the organelle box. If you want it, come talk to me or scan the QR code. We are releasing right now, we started shipping the A549s. The 293s are undergoing QC in the next months, and we hope to release them by the end of the year or early next year. And it takes a village, so thanks to all the team, and I'm happy to take some questions.